Okay. So the objectives for the lesson today: biologists will be able to analyze how mutations affect genes. And the essential question: how can mutations impact a living organism? It is October 13th, 2020. Time's flying. All right. So a mutation is a change in the DNA sequence caused by DNA replication or environmental factors. So sometimes when the DNA is replicated, things go wrong. And ideally, in the process of the cell cycle, during one of those checkpoints, the cell decides not to divide. It decides not to reproduce because the DNA has not been properly replicated. But sometimes these mistakes get through those checkpoints without being detected. Someone remind me, just again, as a bit of review, when does DNA replication take place?
In the S phase? Oh, wait, hold on. No. Wait, yeah, it does.
Yes, during S, during synthesis. And when does S take place? What comes before S?
G1?
G1, excellent, Ayana. And what comes after S?
G2?
G2, good. And so those are the three steps, like Ayana just told us, G1, S, and G2. Those are the three steps of interphase. So sometimes those mistakes happen during replication, but oftentimes they can be caused by environmental factors such as solar radiation from the sun, carcinogens like nicotine, which you get from cigarettes, as well as Juul pods, and other chemicals. Mutations that are found in gamete cells can be inherited. What is a gamete cell? Does anyone know what a gamete cell is?
Sex cells.
Good. Gamete cells are sex cells, egg and sperm. So if the mutations are found in those sex cells, then of course they can be passed on to offspring. We're going to talk much more about how to determine the likelihood that an offspring develops or is affected by a mutation in the next unit. You all will learn how to use what are called Punnett squares to do that. But oftentimes it is not a guarantee that your children or that a child and offspring will be affected by a mutation. The likelihood may be increased depending on where the gene is located, but it's not always a guarantee. In fact, it's almost never a guarantee. Okay. So there are really two main classes of mutations. The first type is called a point mutation. And so when we think about the sequence of DNA, we've got all these different, I don't like that language. Hold on. Sorry if you already wrote that down, but I don't want to say amino acid. I want to say only one nitrogen base is affected. So in a point mutation, only one nitrogen base is affected. So that means that that nitrogen base that should be a T in this case is actually becoming a C. That's a point mutation. But frame shift mutations end up affecting the entire sequence after the point of issue. So there might be an insertion in which we put in a new nitrogen base, but that of course shifts everything else over. Or there's a deletion in which we take out one of these nitrogen bases, and that also shifts everything else over. Okay, so we got to keep in mind that this DNA is eventually going to be transcribed into mRNA, and that mRNA will eventually be translated into amino acids. So if you change the DNA, you have the potential to change the amino acid. Okay, so let's think of it this way. So let's say that I have this sentence: the fat cat ate the rat. Now, does anyone know why I chose words of three here? Why have I chose words of three letters? Hello?
DNA.
What about DNA? I think we should look back in our notes to Thursday or Friday of last week. This is definitely something you all need to know. The question is, why have I chosen words of three letters in this sentence? Someone's got to look back in their notes for me, guys. I know you all have this information.
It would be like a code.
Gosh. Yes, it's called a codon. Yeah, so groups of 3 nitrogen bases are called codons. And it's not necessarily DNA that we're thinking about. It is mRNA. That's why we have those codon charts for the 3 letters. So 3 letters, 3 nitrogen bases represents a codon, and each codon codes for a specific amino acid. So obviously these are not the nitrogen bases. These are not A's and U's and C's and G's, but I did want to just kind of use a simpler example of what we're talking about with these mutations. So let's say that I just change one letter here and I just replace this R with a B. So now my sentence is the fat cat ate the bat. So now the cat might have rabies, but the sentence still makes sense. Okay, the fat cat ate the rat. If I just do one, it's called a point mutation because I'm only changing one of the letters. So the sentence still makes sense, at least. The fat cat ate the rat, or the fat cat ate the bat. It changes the meaning maybe just a little bit, but the sentence still makes sense. All right. Now let's say that I change the order of one of these. Let's say that instead of 8, I change the order to eat. And now my sentence is the fat cat eat the rat. So again, I've changed the order here. So I affected more than just one letter, but the good thing is I only affected one codon. So my sentence is now the fat cat eat the rat. It's not grammatically correct. It's not exactly the same as the original, but it still makes sense. But now let's say that I insert a letter here. Oops. No. Let's say I insert a letter here. I'm going to put an S there now. But keep in mind, codons are made up of how many letters? How many nitrogen bases are in a codon?
Is it 2?
Yes. No, not 2. Not 2.
I think it's between 3 or 4.
It's 3. So okay, come on, guys. Where are our notes? Hello, wake up, wake up. A codon is a group of 3 nitrogen bases that codes for a specific amino acid. Please write that down if you have not already. That's really important. A codon is a group of 3 nitrogen bases that codes for a specific amino acid. Okay. Now back to my sentence. So now that I've put in this S, I still have to read things in groups of 3. That's what my codon says. So what I end up with is the fat ska T to ERA. Does that make sense? Do we see how everything gets shifted over because of that? And so now I don't have an issue with just one codon. I've got an issue with, let's see if I can do it this way. So now my issue isn't just with one codon, it's with 1, 2, 3, 4 codons. They're all messed up because I put in a new letter right here. I still had to read in a codon. I still had to read in groups of 3. So now this becomes ska instead of cat. This becomes T instead of eat. This becomes TTH instead of the. This becomes era instead of rat. So the sentence doesn't make sense anymore. I started off as the fat cat eat the rat, and I became the fat cat ska t to era. Oops, I didn't mean to go full screen. Now let's say if I did the same thing, but I deleted a letter. So let's say I choose to delete that letter E. So now I would end up with THF, ATC, TE, ATT, HERAT, something else there. So now I end up with THF, ATC, 8, at, her, at. So again, when I delete a letter, like I did there, or when I insert a letter, like I did here, everything that comes after it gets shifted over. So this is why we call them frame shift mutations. They shift everything else over. Okay. So the first type that we talked about was a substitution mutation. That's when you only mess up one nitrogen base. And in this case, it may or may not even have an effect on the amino acid. You could still end up with the same amino acid in that case. You could end up with one that is different, but you might end up with the same one because we know that multiple codons often code for the same amino acid. We saw that when we practiced with the codon charts last week. So a substitution mutation is when the incorrect nitrogen base is placed into a DNA sequence. So you put a T instead of a C, or a G instead of an A, or a G instead of a C. Okay. But like we just talked about, when you insert a nucleotide, it shifts the entire frame over. It shifts your reading frame. So we have to keep in mind that we're reading in groups of 3s called codons. So every amino acid that comes after the point of insertion is now going to be potentially messed up. So insertion and deletion are really bad. A deletion really functions in the same way. A deletion also shifts the base over. Or shifts the frame over, I should say. All right. Now, mutations can sometimes be harmful, beneficial, or neutral. Neutral meaning that they don't really have an impact on a person or an organism's ability to survive. Harmful mutations, of course, make the likelihood of survival decrease. So some genetic mutations make life less likely or more challenging. Some actually make life, some are actually adapted. In fact, most adaptations are a result of mutations, genetic mutations, and they make life a bit easier for some organisms. So people with sickle cell disease are resistant to malaria. If we think about where malaria originated in Western Africa, this is a reason why why Black people have a higher incidence of sickle cell, because our ancestors, Black people's ancestors, were more likely to pass on the sickle cell trait because it made them resistant to malaria, which was a big problem where they lived.
I have a question.
Yes.
So you know how like people can be born with an extra limb? Is
Yes.
that harmful? Is that supposed to be harmful or beneficial or neutral?
I think most people would say harmful. I think I'm not sure that I'm aware of many cases of people who are born with extra limbs and they have full control over the limb. Oftentimes they're born with an extra limb and they don't actually have the ability they don't have any motor skills in that limb. So
I met
in that case, it would.
my old teacher. He had an extra finger that he can control.
Yeah.
On his hand.
So sometimes you will see polydactylism, which is having an extra finger or an extra toe. And people who are born in that case sometimes do have the ability to control those fingers, but to control an entire extra arm or limb, leg, I'm not sure that I've ever seen that.
Isn't sickle cell still bad though?
Yeah, sickle cell does still have pretty negative effects. It means that your blood doesn't carry oxygen as well as it could. But what we're saying is, in fact, I don't like the way that this slide is worded either. People who have the sickle cell trait are malaria resistant. That doesn't necessarily mean that they need to have sickle cell disease. So just because you have the genetic marker for sickle cell doesn't necessarily mean that your phenotype, your actual body, produces these sickle cell blood cells. So just because you have that genetic marker doesn't mean you actually get the disease, but even if you have that genetic marker, you will be resistant to malaria. So that was a good question. Thank you for clarifying that. Okay, let's play this quiz. Okay, this is all about DNA and DNA replication. Is this the right? Hold on, I'm not sure if this is the right. I don't know how that happened. Don't join that game. This is the game you should join. So the code is 980778. I'm also going to send a link to the chat that you can click on. So let's not take too much longer to join the game, please. I've only got 5 people. There are 11 of us on the call. I've got 6. Thank you to the 6 of you who joined. 7. Okay. Okay, so we still have some folks who are finishing up, a few of you who are finishing up. That's no problem at all. I will just tell everyone else to go ahead and knock out the exit ticket. And for the asynchronous assignment, just please make sure that you have completed the gizmo, because I see quite a few of you have not done that. So please make sure you take time to complete. There are 2 gizmos. Both of them can be found on the Gizmo website. Yes, I only see that one person has done it. Some of you still have not even joined. It looks like. Actually, no, it looks like almost all of you have joined. But you all need to make sure you go in and complete those assessments for the gizmos. I see that Alicia has done it, so thank you, Alicia. Everyone else, that's how you should probably spend your time.
Is it 11:35?
Yes, it is. And you all are good to go. So please, like I said, make sure you are making progress on those gizmos. They shouldn't take too long. I don't want you to let them back up on you. And I will talk to you all tomorrow.